So the two different types of gene expression controls that we just covered, operons and epigenetics, those are pre-transcriptional control methods. So pre means before. Transcription is, transcription of DNA into messenger RNA, right? So before transcription controls, that's what we covered in the past. That means that was control and gene expression at the DNA level. That's, you're controlling whether the mRNA transcript even gets made or not made. So, and again, if you don't remember transcription and what I mean by that, you need to review you need to go back to the transcription and translation videos that I made for those who need to catch up. So, so operons and epigenetics, that's how bacteria can control gene expression before a messenger RNA transcript is even made. Post-transcriptional controls is what we'll cover now. So what if the cell makes all of these messenger RNA molecules and they're all in line to be translated into protein, and then the cell's environment changes and the cell's like, oops, never mind, I don't want to make any of those. Not only do I not need to make those proteins anymore, I really just don't want to. If I make those proteins now, oh, that's going to be bad. I better stop the assembly line. I have to stop the translation process. The transcripts are there. They're already made. So yes, the cell goes and turns off translation of the gene, or excuse me, the cell will turn off transcription of the genes, but there's a whole bunch of transcripts floating around in the cell waiting to get to the ribosomes for translation. The cell needs to destroy those, okay, so that those proteins are not made unnecessarily. So that's what I mean by post-transcriptional control. This is after transcription. Control of gene expression after transcription has already taken place. You've got the transcripts, you don't want them anymore, what do you do with them? We're only going to cover one, and that's microRNA. So be careful here. MicroRNA, that's different than mRNA for messenger. This is MI and this is M. MicroRNA is a teeny tiny segment of RNA that's complementary to a small segment on the starting end of the messenger RNA, the 5' end, where it fits into the ribosome. So this microRNA, it's much smaller than the messenger RNA. It's a teeny tiny segment that's complementary to the front end of the messenger RNA. When they bind together, the messenger can't get into the ribosome. Now the ribosome can't read it, transcript can't be made. Done. So now this transcript has been inactivated by the binding of microRNA to it. So the whole microRNA, so we talked about all these different kinds of RNA. We have messenger RNA, which is the photocopy of the blueprint of the protein we want to make. We have the ribosomal RNA, which makes the ribosome. We have the transfer RNA, which brings amino acids in the correct order to the ribosome. So all the RNA that we've covered so far, they all assist the production of protein from the DNA code, right? They're all important. MicroRNA stops the production of proteins from the DNA code, okay? This is a negative control as opposed to a positive control. And so it still is involved in controlling gene expression, right? It's just a negative control on gene expression instead of a positive one. So once microRNA binds through complementary base pairing with the messenger RNA cannot be translated by the ribosome, and actually usually the microRNA also attracts enzymes to destroy the messenger RNA. So they're enzymatically targeted and destroyed, these messenger RNA transcripts that have the microRNA attached.